Okay, good. So welcome to our second day of class, where we will start really talking about some very substantive material. I hope all of you so far are having a good week. I wanted to remind all of you that you have recitation this week. Your first meeting will be on either Thursday or Friday in your recitation section, depending upon what section time that you signed up for. And hopefully you're all looking forward to meeting your teaching intern who will be leading you through the semester. I know all of them are very excited to meet you as well. This week in recitation, you'll get used to how the recitation works. You will also have a practice quiz so that you can see how the quizzes are going to work during recitation. For that reason, it's very important that your computer, your laptop or your tablet is able to handle both having Zoom live with your camera on during recitation so that your TI can see you, and have Laulima running at the same time so that you can take the quiz when she or he opens the quiz up to you. Okay, so I think you're all aware of that, but just a heads up that you need to be able to do that for recitations to work. Okay, so let's start by talking about some basic chemistry of the cell. I think a good way to start off thinking about this is because this whole semester we will be talking about complex molecules that are inside of the living cells in your body. It's good to kind of get a little perspective on the size scale of the things that we will be talking about. So in science, we typically use the metric system, and I think, basic unit of measurement that I think you are all familiar with is a meter, right? So a meter is very comfortably on the size scale of what we deal with every single day in our daily lives. In many ways, that's kind of like what we've evolved to kind of be sort of familiar with, because that's what we see around us. So a meter, it's 39.4 inches. It's a bit longer than a typical adult arm, right? So if we start from a meter and we break it down and we break it into 100 equal divisions, each one of those is a centimeter, right? So a centimeter can be written as 0.01 meters or 1 × 10⁻²meters. And a centimeter, I think we're all still very comfortable with that. A centimeter is about the diameter of something like a pea. Right? If we take a centimeter and break it down further into 10 divisions, each one of these would be a millimeter. Right? So a millimeter then is about the width of something like a coin, pictured here. So we're still in this comfortable range of what we can kind of see with our eyes, and therefore what we're sort of familiar with dealing with every day. But if we're going to talk about cell biology, we have to go smaller. So if you take one of these millimeters, right? So keep in mind that one of these millimeters is already 1,000th of the starting meter. And you take one of those millimeters and you break it down into 1,000 more individual equal divisions. Now you're down at the level of a micrometer or a micron, right? So 1 micron is 1 micrometer, with the little mu here, the Greek symbol. There are 1,000 little red dots here, but really what I've done in order to get them to fit on the slide is sort of like layer them up. If you were to string all these dots from left to right, that's what we're sort of talking about as being a micrometer. So that's the scale of cellular biology. Right? Because a typical cell in your body is in the size range where micrometers is the best sort of unit to measure them. So a typical cell in your body might range anywhere from 5 micrometers in diameter to 100 micrometers. I use the word typical cell, right? Because of course there are examples of cells that are smaller than that and way larger than that. So as an example of a typical cell, we have a white blood cell here. A white blood cell that's floating around in your circulatory system, that's helping you fight against infection, is about 7 microns or 7 micrometers in diameter. Right? So that's kind of typical. Right? An example of a atypical or, kind of extreme cell is something like a neuron. So neurons can get up to around a whole meter in length. So for example, the nerves that run from the base of your central nervous system, your spine, all the way down to your feet, those are one continuous neuron cell, starting from your spine, ending down at, let's say, the muscles in your foot. Right? Now, the cool thing about that is that you would not be able to see that with your naked eye because it's so thin, right? And yet this meter-long thing that you can't even see with your eyes, right, is one cell. So that kind of gives you a sense. I would like you in this class to be familiar with these different size kind of scales. I think all of you already knew like what a centimeter is in relation to a meter and a millimeter. Maybe you're already familiar with micrometer or even smaller, nanometers, right? So hopefully this is just review, but it's a good way to start off thinking about this. So let's look at this chart here. If we start on the biggest end of this chart being 100 micrometers, so already very tiny, right? So typical eukaryotic cells are somewhere around this range from 10 micrometers, greater, right? If we were to look at things like prokaryotic organisms, for example, bacteria. So here's a bacteria here. Most bacteria are anywhere from a fifth to a tenth the size of one of your typical cells in your body. The organelles that make up one of these cells of your body, things like a mitochondria, for example. Here's a mitochondria. Now you're getting down to maybe like just a few microns in size. What about things like viruses? So viruses are things that I think are very sort of like at the forefront of our mind these days with the whole COVID-19 pandemic, and we worry about the SARS-CoV-2 virus. How big is a virus? Viruses are actually tiny, even compared to a typical cell in your body. You can see viruses are down in the nanometer size range, quite a bit smaller than the cells in your body that they actually infect. Even smaller still are different protein complexes that are found inside of your cell. These are the protein complexes that make up things like the organelles. And importantly, these are protein complexes that are in the cytoplasm of the cell doing various functions. Right? Remember, I showed you a picture during our first lecture of an amoeba. And it's kind of a neat picture. Inside the amoeba, there's all these different specks and dots and things. And I was kind of joking. I said, that's not because like the lens of the camera was dirty, right? Or you cannot resolve a high resolution image. Those are different complexes, large complexes of different molecules together doing things, right? So those different complexes like ribosomes, for example, right, are very tiny, maybe like just over 10 nanometers. Once you get below 10 nanometers, now we're talking about things like individual proteins or the lipid molecules that make up the membranes of your cell, or things like DNA and RNA. Right? Below that, you get down to small molecules like water, which is much smaller than even a nanometer, and then down to the level of individual little atoms. Okay. So I think this table is another good reminder to you that this is sort of like the scope of what we talk about in this class. When we talk about biology in Bio 275, we start from the level of atoms, simple molecules, complex molecules getting bigger and bigger, up to things like organelles. And then finally, we stop with the living cell. Okay. So let's start then from the very bottom. Let's talk about atoms. Right? So carbon is the most important atom in biology. I think you know this because you spent all of a previous semester studying organic chemistry, and organic chemistry is the study of molecules that contain carbon. right? Now, many of the molecules that you studied as part of organic chemistry may or may not actually be relevant necessarily to the things that we are talking about in a living cell. Right? But in the very least, the things that you've learned about how atomic bonds form and different functional groups coming together to form bonds is relevant. Okay, now I can already tell many of you are cringing. You're like, oh, you mean there actually is some organic chemistry in this class? There's not a lot of organic chemistry in this class. Okay, so if you had a hard time in OChem, don't worry. Okay, but carbon, carbon has 6 protons in its nucleus, 6 neutrons, and as a result, because it has 6 protons, it has 6 electrons, right? And together you have a carbon atom. So this is the old-fashioned model we call the Bohr model of how atoms are originally kind of visualized, right? Sort of like little solar systems. I think even when I was a little kid, most of my little books on science and things like that, even maybe when I got up to high school, they still kind of portrayed atoms like this, right? And this is how the general public thinks atoms are, like little solar systems, right? But that's wrong. The reality is, and again, hopefully you guys already know this, is that electrons are not in orbits like planets around the sun. They occupy these different three-dimensional spaces. They are in clouds of probability. What do I mean by probability? That means within these defined spaces, electrons, right? Electrons can be anywhere within these orbits, right? So here is sort of our modern quantum view that's more realistic of what an atom looks like. And it's how I would like you to think about atoms when we talk about chemical bonds. Right? So we have these different orbitals. We have these lowest energy sort of spherical orbitals where the electrons in the 1s, 2s orbits can occupy. And then beyond that, once you have more electrons all sort of in there, keep in mind, right, the electrons don't just float away because they're negatively charged and they are attracted to the positive charge of the protons in the nucleus, right? So the electrons want to get to the nucleus, but they hate each other, right? They hate other negatively charged electrons. And so they all kind of are stuck in a way in these orbital spaces. Right? So once you start having more electrons that are part of this atom, right, the space that they can occupy becomes more and more defined because they want to stay away from each other. So you begin to have these kind of hourglass shaped or dumbbell shaped orbitals, right, as more and more electrons squeeze in. So valency is a term that you've all studied from general chem through organic chem. It has to do with understanding what electrons in a given atom are actually available to participate in binding 2 atoms together to form a covalent bond. Right? And the electrons that participate are the ones that tend to be in the outer orbitals. Right? So valency can be thought of in 2 different ways. You can either think of valence number as the number of electrons that are occupying the outer orbitals of an atom, right? And that way, the electrons that you sort of picture are already there as part of the atom before it's interacting with another atom. Or you can also think of valency as the question of how many additional electrons, right, should come in to help stabilize the electrons of that atom. In other words, how many electrons are needed to sort of fully complement and fill out those outer electron shells so that the atom is happier. I tend to think it's more intuitively useful to think of valency as how many electrons are needed to finish the electron, the octet on the outside. So therefore, I think of it this way. Carbon, it has 4 valence electrons, and it can take 4 valence electrons in order to stabilize its outer shells. Hydrogen has a valency of 1. Oxygen has a valency of 2, right? Because there are 6 valence electrons already present. Therefore, 2 electrons from someplace else, like another atom coming along, can come in and help stabilize the octet. Nitrogen, right, has 1, 2, 3, 4, 5 valence electrons, and its valency then can be thought of as 3 electrons need to come in, so you have a full 8. All right. So therefore, it's the interaction of these different electrons from two atoms coming together that form bonds, right? So bonds between, for example, carbon and hydrogen, so that the carbon gets the 4 valence electrons that it makes happy, it makes it happy. Each of the hydrogens get the 1 that makes it happy, so it has a pair, right? Methane is CH4. Ethanol, you can see here, right? Here's an oxygen group that's getting its 2 valence electrons contributed by this hydrogen and this carbon, so it has a full 8 now. Methylamine, right? So it's a methyl group with an amine group here, right? And so this nitrogen is happy and these hydrogens are happy. Now, this is an interesting thing to think about, right? Methane is natural gas. It's the gas that's used to burn on gas stoves, for example. So you're sort of familiar with its properties. Ethanol, right, is the type of alcohol that we drink in beer and wine. Methylamine is a compound that is kind of notorious because it can be used during the production of drugs like illicit drugs like methamphetamines. Right? But if you actually look at their composition, right, they're not that different. So the ethanol has an oxygen here, right? It's kind of like it's 2 methanes almost with an oxygen stuck to it. A methylamine is a methane with a nitrogen stuck to it, right? So when you look at them, they look very similar, and yet they have profoundly different properties, both in terms of thermodynamics and how our bodies interpret them and the effect that they have on the cells in our body, right? So how could that really be, right? I mean, after all, these are just different combinations of letters, C's, H's, you throw in an O in one of them, you throw in an N in one of them, right? How could they actually have such profound differences? So that's one of the questions that we'll be studying this semester, and this idea that it's not so much about the fact that there's an oxygen or a nitrogen, it's what this impact has on the shape of this little molecule. And in cellular biology, it's all about shapes. Right? So let's just kind of wrap up this slide by saying that the formation of these bonds then, right? Why do these atoms want to form bonds? They want to form bonds because by sharing electrons, they help stabilize their electron clouds. Think about it this way. Let's say you go to Party City to buy some balloons, right? And you're walking out of the store and you have one balloon on a string and you're walking to your car and it's a windy day, right? That balloon's like flying all over the place and you're struggling to kind of keep it under control, right? But if you have a whole bunch of balloons and you're holding on to them in the same spot like this, right? Ironically, you might think the more balloons you have, the harder time you have keeping this thing under control. But in reality, the more balloons you have, the more you fill out sort of this shape in your hand, right? Where these balloons are repelling each other. And so they more happily occupy their space and they're just kind of, knocking up against each other, but you have an easier time carrying them, right? So just like the more balloons that you hold, the less wild each one flops around. And we're going to kind of use that analogy to think about the energy of these electrons that are occupying these clouds. So how should we think about the energy of chemical bonds? So here's a little story to help us think about it. Right? Because, when we talk about electrons, that's down at such a small scale, it's hard for us to really grasp, right, any kind of intuitive way of thinking about electrons and bonds and things like that. Hey, that's why I had such a hard time in chemistry and OChem. Right? To me, it was all letters and sticks, and I couldn't figure out what was really going on. Let's imagine we have a friend here, Valencia, and she is a waitress in a very busy restaurant. Right? So she is an electron as part of an outer orbital in a particular atom. So she has to cover the entire floor of the restaurant by herself because maybe that day her coworkers called in sick or whatever. Right? So the amount of energy associated with Valencia doing her job is very high, right? She's frantically going around table to table and there's just way too much work for her. If there was another server helping her out and sharing that same floor with her, that would really kind of make Val's own energy requirements a lot lower. So let us imagine that we have a chlorine atom here. Let's say the chlorine atom on the right, okay? And so that's why the the electrons for that one are in orange. And let's just say that Val is this electron here, right? So Val had to cover the entire orbital space that she's part of all by herself. But if another electron came in and shared that space with her, that makes things a lot better. Right? Val would be a lot happier, right? And as a result, maybe this other electron is the same outer electron on a different chlorine atom. That way, these two chlorine atoms come together and form Cl2, right? Because now these electrons are sharing that same orbital that they're part of. So each of the energy that's associated with each of those electrons is quite a bit lower, and things are more stable. But the truth is, Val, this is still a very busy restaurant. She'd rather just go and work like at a mellow cafe somewhere, right? So, for example, there's a cafe that opens up down the street. It's this little French bistro, right? And she likes it because it looks all nice and elegant inside. But more importantly, it looks very mellow and relaxed. She'd rather work there, right? Because then it would be a much lower associated energy job. But this can only happen. In other words, Val can only go and work there if two things happen, right? Number one, that a better job is available in the first place. Right? In other words, there has to be another atom that comes along, in this case, a hydrogen atom, right, that offers a lower energy orbital for Val to potentially participate in. Right? And the other key here is the electron has to be motivated to actually leave its original orbit and reform a lower energy orbit. So this is like Val, and I'm sure you've all had friends like this. Your friends who complain about their job. Oh, my job sucks. It's so busy. I hate my job, right? And then you tell them, you're sick of hearing this from them, right? You're like, all right, whatever. You should quit your job already. Why don't you just quit, right? And then your friend is like, no, it's actually all right, I guess. Right, okay, right? They need motivation. So not only do you have to have a better job come up, here's like the mellow cafe, right? You actually have to put some energy into this to actually boot Val, the electron, out from her original orbit in the Cl2. And then she has the opportunity to kind of look around and take the better job. And the better job then is now the formation of HCl, right? So now she's participating with the orbital of the hydrogen here compared to the shared orbital with the other chlorine. This is a much more chill kind of orbital, right? So this is favored, right? And now she saves a lot of energy. So you got to put some energy in just to break that chlorine atom in the first place, because otherwise it is stable. Just like Val in her original big restaurant job, right? The reason she doesn't want to leave in a way is because at least it's a stable job. This restaurant's not going anywhere, but she would rather be in a much more lower energy state. Okay, and that's then presented by now the formation of HCl. So the question is, what happens to that difference in energy? Val was always very frantic running around the restaurant in her first job. Now look how happy she is, right? She's super happy in her new French bistro. What happens to that difference in energy? That energy is not necessarily lost in living cells, but it can be used for other things. So in this case, Val has free time. She can do some other things, right? Go for bike rides, whatever. She's much happier. In biological systems, the energy that's released by electrons going from higher energy bonds to lower energy bonds can be harnessed by other molecules that are there, ready to receive that energy, and perhaps use that energy as the motivation for them to rework their bonds. Now, if you don't have other molecules ready to take it, then what does happen to the energy? Then the energy actually is lost because it's just released, right? And it's released as heat. So let's talk about this idea of releasing energy as heat, because this is a good way of thinking about, the change in energy associated with bonds. So we'll start off with simple probably like the simplest type of organic compounds, which are hydrocarbons. So hydrocarbons are nothing more than C's and H's arranged in different numbers and different kind of shapes, right? So we already said methane is CH4. Here are several different images of different ways of looking at a molecule, visually represented. It's kind of weird if you think about it, right? Because the molecules are so small, and the reality is they don't really look like anything. I mean, you have like, what, 5 atoms. What does that really look like in real life, right? In fact, they're so small, you might not realize this, they're so small that you wouldn't even be able to like see them as anything, because they're so small that the wavelengths of light are even much larger than the atom itself, right? But we try our best to come up with different illustrations so that we can intuitively imagine what we're thinking about. And unfortunately, a lot of times we get too caught up in these illustrations and we think that molecules look like this, right? For example, we think that molecules look like a C with lines, little sticks connecting them to H's, right? Well, you can get a little bit more accurate because, as you know, the H's are kind of repelling each other. So instead of being flat, like on a piece of paper, right, they would naturally assume these different bond angles. So that's what gives the methane more of a pyramid kind of shape. We can say, let's fill it in with little models, right? So this is kind of typically like when you have your OChem little kits, little tinker toy kind of things you put together, right? But an important thing to keep in mind, right, is all of these always represent the bond, either as a little stick, right, or the little part that snaps together on the model, right? And that is very misleading because the reality is, right, there is no stick that keeps the hydrogen and the carbon together, right? Their electron orbital clouds are fused together. Right? So it's way more accurate to think of the shape of methane as something like this, right? You get rid of the sticks and you really appreciate the fact that they are sharing their electron clouds, right? And therefore, the resulting three-dimensional shape of this, right, is simply the shape that's defined by the electron clouds of these atoms. So here are some examples of some simple hydrocarbons. Here we have ethane, propane, ethylene, acetylene, right? Just variations of C's and H's with different bonds. So here's a question, right? If they're all just C's and H's connected and overlapping in different ways, then why is it when you burn something like methane, and it's nice and hot to cook your food, for example, right? Why does methane burn at a temperature that's lower than something like acetylene? Right? So acetylene in a welder's torch burns a lot hotter than a stovetop. Right? A welder's torch has to melt metal, right? Obviously, the methane on your stove is not melting the metal of your cooking pot, right? But why does that happen? Here's methane, here's acetylene, right? What's going on? So let's think about it this way. Here is methane reacting with oxygen, and the electrons in each will have an opportunity to rearrange so that they can then assume lower energy configurations, resulting in carbon dioxide and water, right? So this is combustion. So the question is, in the process of burning methane, where in this equation, the left side of the arrow or the right side of the arrow, where is the energy actually released? So if this were a normal in-person class, I'd give you guys a moment and I'd sort of have you shout out some answers here. But let's just kind of, for the sake of time, move on. I always thought when I was learning chemistry, like in high school and college, whatever, that it's when you break these bonds on the left, right? You break the bonds and energy is released, because we were always told that bonds store energy, right? But you know what? I hate to break it to you. That's wrong. Okay. Energy is actually released not by the breaking of these bonds on the left. Energy is released upon formation of the bonds on the right. Okay. Energy in this case as heat is released when carbon dioxide and water form, not when methane and O2 bonds break. Right? Why is that? The reason is this. Think about the example of Val the waitress again, right? Val that's in a high energy job. She wants to get to a lower energy job, right? You can say, well, when she finally leaves her high energy restaurant job, how much energy is freed up? And you suddenly realize, well, that all depends. It all depends on what is the next job that she's going into, right? Because until you know that that next job that she goes into is that mellow French bistro cafe, only then do you have something to compare it to. And only then is it relevant or meaningful to say, how much free time has Val gained, right? Just by leaving her first job, right? You don't know what that energy is that we're talking about, because the assumption is she has to have a job. She's not going to just go unemployed, right? So it only makes sense to wait to see what the new job is, and then you know how much energy is liberated. Right? So of course, in a reaction like this, again, you need to motivate the electrons to break in the first place, right? So methane and atmospheric oxygen, O2, is very stable. Right? These molecules can exist for tens of thousands of years just floating around, right? So you need to motivate them to break their bonds. And of course, that is heat, either in the form of a flame to start, like your acetylene torch, right? Or the little electronic spark starter on your stovetop, because that's what gets the electrons to separate. And when that happens, the electrons have an opportunity to see that there are better arrangements for them to reform, and then the energy is released. So bond formation then is a downhill flow of energy. What is energy? Energy is the ability to cause matter, right? Atoms are the basis of matter, to move. And also the resulting movement of matter is in itself a form of energy. So if we think about this example of a large boulder or a large ball on the top of a hill, we all intuitively know that, yeah, the ball in a weird way would rather be at the bottom of the hill, right? There's that potential energy there. But that ball could stay on the top of the hill indefinitely, 10,000 years, something like that, right? Kinetic energy then, that potential energy gets converted into kinetic when some energy is put into it to push the ball and get it going. Right? So as the ball goes down the hill, that energy is then released. So you could use it to harness it and do something else. So you can imagine, for example, let's say there are lots of things on this hill that you want to knock down, right? Then you can say that energy is being used now to do other work, like knocking these things down. Or if there's nothing and the ball just ends up at the bottom, then the energy is kind of like wasted in a way. It's just released, right? But the point is, it takes energy to break these bonds in the first place. So let's compare then the methane with the acetylene and see why there's a lot more heat involved with the acetylene torch. So the methane reaction, this is the one we looked at on the previous slide. We have 2 methanes reacting with 4 oxygens. You give it some heat, which is the same as putting in some energy to get these electrons in the methane and the oxygen to actually get disrupted from their orbits, and they rearrange, right? So in this case, the oxygens rearrange with the carbons to form 2 molecules of carbon dioxide, and the oxygens rearrange with the hydrogens to form 4 molecules of water. Therefore, all of these bonds, the electrons that are on the right side now, are in a happier kind of state, right? And that difference in energy is now released as heat. Let's look at acetylene. 2 molecules of acetylene, just like 2 molecules of methane here, right, will react with 5 molecules of oxygen. And when they reform, they will reform as 4 molecules of carbon dioxide and 2 of water, right? So the important thing here, which I think is already apparent to you, is in both of these situations, if you count up the number of red, black, and white balls on the right side, it's the same as on the left, right? So things are being perfectly rearranged. But notice that with the acetylene, there are a higher number of more stable bonds that are formed between the carbons and the oxygens here, right? And as a result, there's a bigger drop in energy from start to finish. And if that bigger drop in energy is also being released, that means it's more heat, right? Okay, so combustion of acetylene yields more lower energy bonds, and therefore more energy is released to either move other molecules or just lost to the environment as heat. So another important concept with molecules related to life of living cells is the importance of water. So water makes up about 71% of the Earth's surface. Anything from 75 to 85% of a living cell is actually just water. And in fact, 60% of our entire body weight is due to things like water, right? So life on Earth requires water. What are some of the properties of water that make it so important for life as we know it? So water molecules are polar and they are cohesive. Polar means, right, when you have an oxygen with two hydrogens attached to it, the oxygen tends to be greedy with the electrons that are being shared with the hydrogens. It has more pull, right? I mean, you can imagine that because the oxygen is a larger atom to start with, right? There are more protons in the nucleus compared to the sad little one proton in each of the hydrogens. So as a result, even though they're sharing the electrons, it's not an equitable sharing, right? The oxygen tends to pull the electrons more towards it. So that means the water molecule tends to have a negative charge associated with the oxygen end and a positive charge associated with the hydrogen end. So that makes it polar. All right, so that's very important. If you look at what happens in water, water molecules then will tend to interact with each other, right? The sort of net negative charge on the oxygen is kind of attracted to the net positive charges of those hydrogens on another water molecule. So as a result, the water molecules kind of form these very weak interactions, right? Because these dotted lines are not actually the atoms sharing electrons in orbitals, right? It's just the attraction of positive and negative charges because of the polarity. So these hydrogen bonds are very important in biological chemistry. They're about a tenth as strong as a true covalent bond. Water also stabilizes temperature. Water is a great temperature buffer. Right? Again, because of the hydrogen bonding network. Think about it this way. When you're heating up something, what are you doing, right? You're putting more energy into something, and that means that all of the molecules in whatever this thing is that you're heating up are beginning to kind of vibrate and become agitated more, right? And that means all of the electrons and all of the chemical bonds are sort of getting more and more potentially disrupted. In water, because of this hydrogen bonding network, those dotted lines, it's like a buffer, it's like a cushion. You can imagine these individual water molecules can still kind of vibrate quite a bit. And they're all just kind of held together by these dotted lines, almost like little springs, right? That can absorb a lot of energy. It's kind of like your car's shock absorbers on a bumpy road. Right? Also, water is an excellent solvent. Remember, solvents are fluids or substances in which you can dissolve things. So those things that you would dissolve in them are called the solutes. So water is remarkable because, again, of this hydrogen bonding property. Let's look at this here. Here, let's say we had a molecule of table salt, sodium chloride, NaCl. Originally, this purple Na plus was attached to this green Cl minus because of an ionic bond, not a covalent bond, right? An ionic bond is when the sodium actually gave up one of its electrons and the chlorine happily took it, right? They are not sharing the electron. It's actually the Na donated the electron to the chlorine, and that made the chlorine automatically, instantaneously negatively charged because it has an extra electron, and then the sodium instantly became positively charged because it lost an electron. And so guess what? The positive and negative charges just keep them happily kind of hanging out together. right? So that's an ionic bond. But water, if you put the salt crystal, let's say in water, these little water molecules, these pesky water molecules start getting in the way, right? And they can actually start separating the sodium from the chlorine because of the polar nature of the water. The sort of negatively charged oxygens of the water molecules happily are attracted to the positive sodium. The positively charged hydrogens of the water molecules are happily attracted to the negative chloride ion. And as a result, right, the salt dissolves very easily as a solute in this particular solvent, water. So substances that interact with water very easily are what we call hydrophilic, right? They love water. Things like sugars, this example of a table salt here, nucleic acids, or proteins that are dissolvable in water, we call soluble proteins, right? They're all hydrophilic. Oppositely, there are molecules that don't have charges on them, and as a result, the polar nature of water is kind of irrelevant. Right? So certain molecules will not form these kind of water shells around them. The water molecules will just keep bumping into them, pushing them around. And we call those hydrophobic interactions, right? And hydrophobic molecules, as a result, tend to separate from water because of all the water molecules just banging up against them and shoving them aside, right? So things like lipids, so fats, right? That's why oils, separate from water very easily, or certain proteins that are insoluble, that don't have charges on the outside, they won't interact very well with water. Right? Another concept is the formation of what we call polymers. So polymers are simply a description of larger molecules that are made by assembling individual repeating building blocks that are smaller molecules. Each of the smaller molecules is a monomer. It is something like a simple sugar or an amino acid or a nucleotide. Right? You can see here's glucose. Right? Glucose can be strung together to form a long chain of sugars such as cellulose. Amino acids that you see here can be strung together to form a long chain that eventually folds up to form a protein. Or nucleotides like A's, T's, C's, and G's, right? They are individual building blocks. They are monomers that can be linked together to form something like a long nucleic acid chain like DNA. Right? So this understanding of monomer building blocks and how they come together to form complex, bigger polymer building blocks is an important thing that we'll talk about over the next couple of lectures. All right. Importantly, your cells do this. Not only do they put together polymer chains, but they also take them apart to get these raw building blocks back out again. Another thing that's important for understanding cell biology is that large molecules will have a tendency to self-assemble. What do I mean by that? Right. In this image down here, you have a polymer chain of something like amino acids that's forming the chain that's going to fold up as a protein. And in this particular case, the chain will itself spontaneously fold up into a particular shape just by the nature of the amino acids and where they are in the chain. If you were to heat this up, again, you start giving them energy so that all these bonds start shaking. Eventually, you can break these different interactions and the protein will denature. It will unravel back to its original linear polymer structure. In some cases, if you cool things down again, you take away all that extra energy that's causing a problem here, that chain might actually collapse again back into the renatured original shape. Okay? And the important thing to keep in mind here is that many times these spontaneous folding or spontaneous interactions of different biological molecules, right, are non-covalent. It's not like all of a sudden different parts of these long molecules are sharing electrons with each other, right? This is just a virtue of things like the shape and also the interactions of water around it, kind of shoving things into a particular shape that minimizes the energy that's associated with the structure. So here, this purple line is our first introduction to something like a polymer chain of amino acids. If you were to fill this out and actually imagine the different atoms that are making up this chain, you might get a structure like this, right? And so this would be an example of a protein. In this case, this is a real protein. This is insulin, right? Insulin that is secreted by your pancreas to help you properly make use of the food that you're eating and the sugars that are part of the food that you're eating. We will study this more as we get into particular examples. So there are four main macromolecule families that make life possible on Earth. So start from the right side of the slide, right? These are macromolecules. They're big. They're made up of different subunits. Polysaccharides, right, are the long chains of sugars. They are made up of individual building blocks that are simple sugars, so glucose and other monosaccharides. Right? Glucose itself, right? It's 6 carbons with some hydrogens, or like an oxygen in there, right? It's made up of inorganic precursor molecules like water, carbon dioxide. Right? Another family are lipids. So lipids are things like fats and oils. Right? Proteins, another big one, we already sort of briefly talked about, are made up of amino acids. And nucleic acids, like DNA and RNA, are made up of nucleotides. So these are the four main macromolecule classes that we will study this semester, and we'll go into depth with each one. Membranes are very important. Membranes define boundaries that give a living cell the ability to be self-contained and have special compartments within it where different types of tasks can take place. So I think you already know from your intro biology that the cell membranes of our cells are a lipid bilayer, right? 2 layers of phospholipids that form this boundary. We will lecture later on in the semester about this lipid bilayer, so we'll go into more depth about it later. I just wanted to introduce that. So to summarize then, there are 5 molecular principles that are fundamental to life on Earth. 1 is the importance of carbon. 2 is the importance of water, right? And the polar nature of water that makes it such a great solvent, for example. 3 is the importance of selectively permeable membranes that actually define boundaries of the living cell. 4 is the importance of forming polymers to make large macromolecules that make many functions of the cell possible. And 5 is the importance of self-assembly, the tendency for large molecules to on their own form certain shapes, and those certain shapes then being the right shapes to interact with other macromolecules. So all of these complex pathways of life that take place in our cell are fundamentally driven by atoms that are seeking stability through the making and breaking of bonds. Okay, so that's it for today. Let me go ahead and stop the lecture.